Hey everyone, welcome back. This is your part 1, chapter 8 notes on genetic inheritance. In this first part, we're going to be looking at Mendelian genetics. So this is traditional Mendelian genetics. And in the second part of our notes, we will be looking at non-Mendelian genetics. A little bit of background first. In the 1860s, the monk Gregor Mendel used scientific method to studies of garden peas. Well, the nice thing was that garden peas were easy to grow and manipulate. Their organs have both male and female sex organs, which are known as pistils and stamens, that will produce gametes. Now, the way that he described it was in terms of their characters. So these are the observable physical features, such as flower color or seed shape. And he noticed that they had different traits. And these are the forms of a character. So for seed color, they could be purple. For flower color, they could be purple versus white. And for seed shape, they could be wrinkled versus smooth. Mendel worked with true breeding varieties. When plants of the same variety are all crossed and all offspring have the same characteristics. They didn't have the terminology that we have today, but this is what led to that terminology. He identified the parental generation as P and any resulting offspring as the first filial generation, or F1. If the F1 plants self-pollinate, they produce the second filial generation, or F2. And so the word hybrid refers to an offspring of crosses between organisms differing in one or more characters. In the first experiment, Mendel crossed plants differing in just one character, producing monohybrids in the F1 generation. The monohybrids were then allowed to self-pollinate and form the F2 generation, a monohybrid cross. One trait of each pair disappeared in the F1 generation and then reappeared in the F2. Mendel's finding led to a rejection of the common idea or common hypothesis of the time, which was called the blending hypothesis. The F1 offspring were not a blend of the parental traits, but only one trait was present. The trait did not disappear in F2 generation, it showed up back again. So in his study, all 7 crosses between varieties of contrasting traits gave the same kind of data. Mendel called the trait that appeared in the F1 generation was more abundant in the F2, the dominant trait, and the other was the recessive trait. The ratio of dominant to recessive in F2 was about 3:1. Mendel proposed that genetic determinants, or genes, which we now call genes, occur in pairs and are segregated into gametes. Each plant has 2 genes for each character, one from each parent. We now use the term diploid to describe having 2 copies of the gene, and haploid as having 1 copy of the genes. Mendel concluded that each gamete contains 1 copy of each gene, haploid. The resulting zygote contains 2 copies, which is diploid, because it is produced by the fusion of 2 gametes. And we know this now as fertilization. Different traits arise because there can be different forms of a gene, now called alleles for a particular character. So everything prior to this had been Mendel's work, and with his limited information about genetics and DNA as the genetic material. Now that we know about DNA as a genetic material, we're going to be using different terms and terminology for genetics. For example, we have the first term, which is alleles. So the different forms of a gene are called the allele. For example, flowers, the flower color characteristic can be in the purple allele, the white allele, red allele, orange allele. So it's describing the same characteristic, but there can be many forms of that characteristic. We also now incorporate the terms phenotype, which is the physical appearance of an organism, and genotype, the genetic makeup of the organism. Capital letters designate the dominant allele, lowercase designates the recessive allele. And for example, round seeds can be the result of 2 different genotypes, big R or little r. Please remember that the term dominant allele, although Mendel had coined it at the time, because of the abundance of the particular allele, we do not use abundance as evidence for something being dominant. It is dominant because it hides the recessive allele, not because it is the most abundant. True breeding individuals have two copies of the same allele. In other words, they are homozygous for the allele. Heterozygous individuals have two different alleles. In other words, they'll have a capital letter and a lowercase letter. This is an example of a cross that Mendel would have performed. So the allele the character he was looking at was the shape or the texture of the seed, and the two alleles are round versus wrinkled, or smooth versus wrinkled. And so you can see here what is indicated as dominant, and as they separate into the gametes, we place these on the Punnett squares to estimate or determine what the probability of the offspring are. But when the offspring of this generation are then crossed, so you can see here now we have two heterozygotes, our alleles split up, and now you have a different ratio for each of the different characteristics. This leads to Mendel's first law. The law of segregation states that the two copies of a gene separate when an individual makes gametes. Each gamete receives only one copy of the characteristic. Gametes from a homozygous individual, in other words, big R, will all be R. Gametes from a homozygous recessive condition will only be recessive R, lowercase r. And their offspring, which is shown in the F1 generation, will all be big R, little r, or what is known as heterozygous. In the F2 generation, gametes will be R or R. So this is, again, because in the F2 generation, they were all heterozygous. So they'll be big R or little r. Genotypes in the F2 generation can be predicted using a Punnett square, which considers all possible gamete combinations. So in this particular one, when we fill out the chart, we would have big R, big R, big R, little r, big R, little r, little r, little r. In other words, 1 is homozygous dominant, 2 are heterozygous, and 1 is homozygous recessive. The Punnett square predicts a 3-to-1 ratio of the phenotypes in the F2 generation. Mendel's experimental values were remarkably close to this for all 7 of the character traits he compared. Genes are now known to be relatively short sequences of DNA found on the much longer DNA molecules that make up a chromosome. So, for example, you may have a chromosome. Okay, you know that in the loose form it's known as chromatid or chromatin, but a gene is just a segment of that DNA that actually codes for a protein. So on the long DNA, you actually may have several genes with what is known as non-coding DNA in between. Today we can picture the different alleles of a gene segregating as the chromosomes separate during meiosis 1. Genes determine phenotypes mostly by producing proteins with particular functions. In many cases, a dominant gene is expressed, and the recessive gene is mutated so that it is no longer expressed or produces a non-functional protein. Mendel verified his hypothesis by doing what is known as a test cross. You're going to cross the F1 with a known homozygote, which has the recessive phenotype. In other words, you're going to take a homozygous recessive and cross it with an unknown dominant phenotype. So the dominant phenotype could be big R, or big R, little r. If the F1 individual was homozygous, all the offspring would show dominant trait. If the F1 was heterozygous, half of the offspring will have the recessive trait. So this is an example of a test cross. We have an unknown dominant condition here, and we're going to cross it with the homozygous recessive. If the individual is big R, then we're going to see different results than if the individual is big R, little r. So depending on what this is, we can determine if its genotype is big R, or big R, little r. Let's see what the results are. So if the individual is big R, then what you would see is all of these are dominant. For example, this one here would be, oops, let's try a different color. Big R, little r, big R, little r, big R, little r, big R, little r. So if all your plants, if all the seeds come out to be dominant, then you know that the unknown phenotype of that parent was homozygous. However, on this one, only 50% of the seeds would come out to be round. So then you would know that the plant is being heterozygous, because the only way that these offspring here would end up being homozygous recessive is if they inherited one of the little r's from the sperm and a little r from the egg. Mendel's next experiments tested whether genes are distributed independently in the offspring. He crossed peas that are differed into two characters, seed shape and color. He used what are known as true breeding parents. In other words, they were homozygous dominant for both conditions. In the F1 generation, that is the offspring of that first cross, you notice that they are all heterozygous. Crossing the F1 generation in all identical, in other words, they were all identical heterozygotes, so they're a heterozygote for R and heterozygote for Y, is what is known as a dihybrid cross. Mendel asked whether in the gametes produced by the heterozygous F1 generation, the traits would be linked or would they segregate independently. Two possible outcomes of the dihybrid cross. If the alleles were linked, the gametes would be big R, big Y, or little r, little y. They would stay together. And as a result of that, the F2 would have 3 times more round yellow than wrinkled green seed. So it would be a perfect 3-to-1 ratio. Again, that's if the big R and big Y always stick together. The second possible outcome, which is known as independent assortment, is that the gametes could be big R, big Y, little r, little y, big R, little y, or little r, big Y. In other words, there is a chance that the big R could combine with the big Y and the little y, and the little r could combine with the little y and the big Y. And in that case, the F2 generation would have 9 different genotypes, and the phenotypic ratio would be 9:3:3:1. So he ran the test, and here were his results. He found that when doing this cross, the next generation came out to the 9:3:3:1 ratio. That means that the traits separated out in the gametes. In other words, just because you have a big R, dominant R, doesn't necessarily guarantee you're going to get a big Y. You have an equal chance of getting a big R and a little y. And so this is known as independent assortment. The alleles of different genes sort independently during gamete formation. In other words, just because you inherit one trait doesn't guarantee you're going to inherit another trait. This law does not always apply to genes near each other on the chromosome, but the chromosomes do segregate independently. We are going to talk about examples of traits or characteristics that do not always follow independent assortment. One reason Mendel was successful was his large sample sizes. By counting many progeny from each cross, he was able to see clear patterns. After his work became recognized, geneticists began using probability calculations to predict expected ratios of genotypes and phenotypes and statistics to determine whether actual results match the predictions. So here's how they used, these are the rules of probability. If you have 2 coin tosses, keep in mind that you have 2 coin tosses, so 2 separate coins, and they're both flipped. Okay, each coin has a probability of landing up heads 50% of the time, because there's 2 sides to a coin. So the probability that 2 coins will come up heads is found by multiplying. This is the probability of the first coin coming up heads. This is the probability of the second coin coming out heads. So when you want to find the 2 combined probabilities, you multiply them together. This is known as the multiplication rule. The probability of two independent outcomes occurring together is found by multiplying the individual probabilities. So in a monohybrid cross, after self-pollination of an F1 heterozygote, the probability that the F2 offspring will have the genotype big R is 1/2 times 1/2. So that comes out to 1/4. The probability that the offspring will have little r is going to be the same. And you can see this by looking at a Punnett square. 1 out of the 4 is going to be big R. Again, keeping in mind that this is the parents are heterozygous. And the probability that the little r is going to come out is still 1 out of 4, because you multiply 1 out of 2, 1/2 times 1/2, and that's how you get that probability. Now for heterozygotes, it's a little bit different. The probability that a offspring will be heterozygous is, keep in mind that it can either be big R, or little r, big R. So in that case, since there's two different ways to get the heterozygous, we use the addition rule. It's the probability of an event that can occur in two or more different ways is the sum of the individual probabilities of those ways. So now instead of multiplying, we are going to add them. So 1/4 plus 1/4 comes out to be 1/2. Now you all will be using Punnett squares to show these different probabilities. So if we take a look at those parents that are heterozygous, big R, big R, little r, you see that mathematically they come out the same. So you have a 1 in 4 chance of the offspring being homozygous dominant. You have a 1 in 4 chance of the offspring being homozygous recessive. But you have a 2 in 4, or 1/2 chance of it being heterozygous. Human pedigrees can show Mendel's laws. Humans have few offspring, so pedigrees do not show clear proportions that the pea plants would show. But pedigrees can still show patterns and be used to determine whether a rare allele is dominant or recessive. The pattern of inheritance if a rare allele is dominant. Every person with the abnormal phenotype has an affected parent, and either all if they're homozygous parents or half if they're heterozygous of an offspring in the family are affected. And so here's what they show you, similar to the pictures we had on your flipped pedigrees. So you can see here's the dominant affected parent who passes it on to each of their children. And then the same thing, now that each of these are affected, they do have a chance of passing it on to the offspring. Keep in mind that for each of these, you can see here that these individuals were homozygous recessive. So let's write little a. So whether this person is heterozygous dominant or homozygous dominant can be determined by looking at their offspring. Because the only way that this person right here, since they are not affected, they have little a. Well, think about where those came from. This one right here came from this parent. So that means this one right here must have come from this parent right here. In terms of patterns of inheritance when it's a recessive allele, affected people often have two unaffected parents. And we call these unaffected parents, we call them carriers. In other words, they are heterozygous. In an affected family, 1/4 of the children of unaffected parents are affected. And again, they are called carriers. And so on this particular tree, what you're going to see is that they indicate someone is heterozygous by half shading in the circle. So this is an individual who has the normal phenotype and is carrying the recessive allele that we're looking at. And so they shade in half of their symbol. Then an individual who fully inherits both copies of the recessive genes is going to be little a, and completely filled in. Thank you all so much for watching. Bye.